Hi, this is Kathy Deeks, and I cannot figure out how to show my PowerPoint while I'm making this video, so I'm just going to do this the completely outdated, old-fashioned way on holding my PowerPoint like this, and then just re-recording this. I don't know where it went. I worked so hard on this yesterday and made the tape, and now we can't find it. So I'm going to discuss how the mechanism of how the AZT drug works on the HIV virus. Okay. Of course, I don't have a copy of this in front of me. So the first thing I want to do is review the life cycle of the HIV and just overview kind of what happens. The entry is binding of the HIV to the receptors on the CD4 cell. The reverse transcriptase is what the virus uses to convert its RNA into DNA. The integration is the virus uses the enzyme integrase to insert its viral DNA into the DNA of the host CD4 cell. The replication is the HIV replication in activated cells, where there's a proviral DNA that's formed by the transcription using machinery of the CD4 host cell to create new viral DNA and translate it into long polyprotein chains, which become the proteins and enzyme parts of the new viral particles. The assembly is the new viral proteins and enzymes move to the outer membrane and assemble into a non-infectious HIV bud. And then the budding and maturation is where the virus bud is released from the host CD4 cell. The viral enzyme protease cuts the HIV polyprotein chains into small HIV proteins to make mature infectious viral particles. So there's a couple of different classifications of drugs that are used to treat HIV, and I had found an amazing article, and I have it listed in the reference overview of antiretroviral agents used to treat HIV. And it goes through all the different classifications and stuff. And so I do wanted to make sure that I mention that because it just reviews. I learned a lot from reviewing that. But here I'm just going to review what's going on here with our with the one we're looking at, the AZT. So let's see here. HIV RNA, like I said, attaches to the reverse transcriptase enzyme, which uses the host cell nucleotides to make DNA using HIV RNA as the template, and that knocks down the viral RNA. Reverse transcriptase sets up for the integration into the nucleus, and the HIV virus enters through the nuclear pores. It doesn't need the cell in order to do its replication. The integrase enzyme slices the new DNA into the host chromosome, and now the viral DNA provirus is transcribed as a cellular gene. And so we have that host RNA polymerase II that's used to replicate it. And the transcription of the gene produces a full-length RNA leading to several proteins which bud out of the cell, and these are cleaved into functional proteins and migrate to the cell membrane. The budding of the virion occurs, and viral protease cleaves and releases reverse transcriptase, forming the virion core. All right, but specifically to AZT, it's a thymidine analog, and it's on the far. Let's see here. it's right here. That's thymidine, and then the middle picture is the AZT. So they look almost similar. And AZT works as a nucleoside reverse transcriptase inhibitor. It undergoes intercellular phosphorylation by host enzymes, where the negative phosphates are added to the protein, and enzyme kinase transfers them to the target, changing the 3D protein structure and function. So reverse transcriptase would normally use the nucleotide analogs, A, T, G, or C, to build the DNA strands, but where the AZT works is that it inhibits this as a competitive inhibitor, and DNA elongation is then terminated after the AZT triphosphate is incorporated. And I will send a copy of these PowerPoint slides to you. Okay, that's all I got today. I gotta get back to work. Get to work here. Thank you.